I want to welcome everybody to the UK Beef webinar series. And it's my pleasure to introduce Dr. Benjamin Kreitz to our webinar today. Dr. Kreitz is the Vice President of Beef Market Development at ST Genetics. And I'm sure Dr. Kreitz looks pretty familiar to everybody because he's very familiar to UK Beef Extension. Ben got his master's and PhD here at UK and was a very important member of our beef extension group for several years. I think, 6 years, Ben?
That was right at 7, or just shy of 7, yes.
Just shy of 7 years. So Ben's been around quite a while, been again, an integral part of our beef extension group. And we're happy to welcome Ben back to present. And what Ben's going to be talking about tonight is his PhD work, where he is going to describe the influence of Selenium on reproduction in beef cattle. So with that, Dr. Kreitz, take it away.
Well, Dr. Anderson, thank you for that introduction and for those kind words, and certainly was very honored and very humbled to share my PhD research with everyone when I was asked to present at this webinar. So I'll go ahead and Dr. Anderson, just want to double check here that my screen is still correct.
Everything looks great.
All right, great, thank you. So as Dr. Anderson mentioned, the title of my topic tonight is Selenium's Impact on Female Reproduction. So I just want to go through a brief overview for tonight. So I'll go through a little bit of some background on selenium and dive in a little on the Corpus Luteum, the uterine function specifically at maternal recognition of pregnancy, and we'll discuss the results of 2 experiments that we conducted, and then we'll wrap up with a summary of tonight. So as we dive into selenium, and interestingly, selenium was actually discovered in 1817 by a Swedish chemist named Berzelius, and originally it was named Selene after the Greek goddess of the moon. And interestingly, and a lot different than what we think of selenium now, Dr. Lemkuhler, is that it was initially considered a toxin until 1957, when selenium was then found to prevent necrosis in rats. And now we refer to or know selenium as an essential dietary micronutrient when included in low levels in the diet. So a little bit about the selenium requirements. And the suggested dietary requirement for selenium, according to NASEM, or what was the NRC, is at 0.1 parts per million. Then we also have a max selenium supplementation rate by the FDA at 0.3 parts per million in feed, with a daily max intake not to exceed 3 milligrams of selenium per head. And I'm sure that many of you are very familiar with some common vitamin mineral supplementation methods. Those including provided by loose minerals in the form of press blocks, liquid supplements, or molasses-based tubs. However, when we think about selenium, and as we cover the United States, there's really areas in the United States, particularly in the northwest corner, but then really in the east and the southeast. And I want to just highlight some of these areas here. So we have this northwest corner, but then when we look at the east and the southeast in particular, these dark gray shaded areas have areas where about 80% of all forages and grains are selenium deficient, where they have less than 0.05 parts per million. And then variable, as we look at Kentucky and Tennessee, it's actually just under half of all of those forages and grains would be have less than 0.1 parts per million of selenium. But these selenium concentrations in these forages and grains really varies across the United States. And interestingly, when you look here, just in the central region of the United States, as it runs from north to south, that these really dark kind of circles are areas where there's actually selenium toxicities, where there's accumulator plants that may contain greater than 50 parts per million of selenium. So certainly faced with a lot different challenges depending on where you're located geographically. There's certainly some problems that are associated with selenium deficiencies in cattle, and research has demonstrated that selenium deficiencies in cattle can lead to decreased immunity, reduced growth rates, and also decreased fertility. So how is selenium commonly supplemented? As Dr. Anderson alluded to, I'll really discuss, and my research focused on the form of selenium supplemented rather than providing supplemental selenium to no selenium provided in addition in the diet. So those two forms really being the organic form of selenium or the inorganic form of selenium. And the two most common forms of the organic forms of selenium commonly found in forages would be selenomethionine and also selenocysteine. The organic forms of selenium are commercially available, and those are available as in the form of a seleno-enriched yeast. And these also may be referred to as the chelated form of mineral and included in the diet in the organic form. But on the contrary, or in addition, when we have the inorganic forms of selenium, those two common forms being sodium selenite or sodium selenate. And the inorganic forms would be the most commonly used form in the feed industry, and simply due to the reduced cost compared to the organic forms of selenium. So if we really now start to dive into some of the effects of selenium on reproduction in cattle, and some of the previous research that has been done, is that when selenium-enriched yeast, so the organic forms of selenium, were supplemented to dairy cows during the pre and postpartum periods, so the periods right before and after calving, that those cows were found to have increased postpartum concentrations of progesterone. And also in dairy cattle, when providing supplemental selenium, those cows had reduced incidence of metritis and ovarian cysts, but they also had increased first service pregnancy rates. And specifically to some research at the University of Kentucky and in the lab with Dr. Bridges, it was found that a 1-to-1 combination of inorganic and organic form of selenium, and throughout the rest of this presentation, I'll refer to that simply as mixed, that the cows were observed to have increased concentrations of progesterone on both days 6 and 7 of the estrous cycle compared to cows supplemented with the inorganic form of selenium alone. So taking this research was really the foundation of the projects that we'll discuss here in just a little bit to try to determine the mechanism of this increase in concentrations of progesterone early in the estrous cycle. So now as we shift gears and start to dive in a little bit into further detail on the reproductive side of things of tonight's discussion, we'll really focus on the corpus luteum. So corpora lutea are actually developed from follicular cells on the ovary. And these follicular cells differentiate into luteal cells. And this is caused by the pre-ovulatory surge of luteinizing hormone, or LH. There's two cell types that make up the follicles on the ovary, those being the theca cells, which differentiate into small luteal cells, and granulosa cells, which differentiate into large luteal cells. This entire process is known as luteinization. And luteinization is associated with an increase in steroid production, but more specifically, a switch from the ovaries producing estradiol to now progesterone, or the structures on the ovary now producing progesterone. There's also a change in the enzymes that are responsible for these changes. And as we look at the image here in the bottom left-hand corner, this is an ovary on a cow. And this structure in blue, or highlighted here in blue, this protrusion is a corpus luteum. And we have our image on the right, which this is a corpus luteum that has been removed from an ovary and has been split open and cut in half. So you can really see this nice bright yellow. And Dr. Anderson, when those individuals were naming a lot of things in reproduction, and corpora lutea is Latin for yellow body. So things make a lot of sense of how they are named. So we really see this very nice, bright yellow structure when we remove this CL from the ovary and go ahead and cut that in half. So what is the function of a corpus luteum? And the major role is the production of progesterone. And we know that progesterone is required for maintenance of normal pregnancy in mammals. I don't want to spend a whole lot of time to go through the steroidogenesis of progesterone, but really just want to spend a little bit of time up here at the top of this figure. So the sources or the precursor for progesterone and steroidogenesis is cholesterol. And there's really 4 sources of cholesterol for the cell to take up to produce progesterone, those being the cholesterol esters, the uptake of cholesterol through both low and high density lipoproteins. But then we also have where the cells undergo a process known as de novo synthesis or biosynthesis of cholesterol. And this area here of this biosynthesis, we'll see, start to see some very interesting things in our experiment. And I apologize that this figure is a little busy, but I just want to take a minute to talk about what does the profile of progesterone during the normal estrous cycle look like. So here on our x-axis, we have our day of our cycle, with day 0 being that day that cow is in heat. And then we also have our y-axis, or the concentration of progesterone, and this is displayed in nanograms per mil. So right when this cow would be coming into estrus, we have the surge of LH, right, which we talked about a little bit ago, was the process of luteinization. So we have the surge of LH, the follicle ruptures and ovulates, and now that follicle begins the process of differentiation into a corpus luteum, or into a CL. So then if we look at this dashed line, is the profile of progesterone. And just a few days after estrus, we see that there is a rapid incline in the concentration of progesterone up until about day 8. And depending on the textbook you read or the image you look at for this figure, a lot of times we'll see that from day 8 to around day 17 that it completely plateaus. But in fact, there still is an increase, but certainly at a much lesser rate from day 8 to day around 17. And then around day 17, all of a sudden we see a rapid decline in the concentration of progesterone. And then the cow is able to resume her next estrus. But we mentioned that progesterone is necessary to maintain normal pregnancy in mammals. So in order for that to occur, there must be something that happens here to stop this reduction in the concentration of progesterone to immediately decline, but to sustain this level of progesterone at a high level to sustain pregnancy. And this is really known as a process that is referred to as maternal recognition of pregnancy, which again is maintaining that secretion of progesterone beyond the length of a normal estrous cycle. And it actually prevents the endometrial release of prostaglandin F2 alpha to inhibit luteolysis. In maternal recognition of pregnancy, in cattle, the signal for that is initiated by the conceptus, and the signal is interferon tau. And depending on who you ask and what papers you read, and it's not a clear-cut answer as to what day this occurs on. Some say it occurs on day 15, some that it starts as early as day 13, but it's most commonly accepted that maternal recognition of pregnancy in cattle is around day 16 to 17 of gestation. So again, this is what is normally occurring. So normally we would have this pulsatile release of PGF2 alpha, which would be responsible for that sharp decline in progesterone. However, when interferon tau is present, being produced by the conceptus, interferon tau blocks this pulsatile release of PGF2 alpha. So we have interferon tau at the timing of maternal recognition of pregnancy, so around day 17. It blocks this sharp decline in the concentration of progesterone, and now progesterone is maintained and is necessary for a successful pregnancy. So very briefly, some of the effects of the early luteal phase progesterone concentrations, and research has demonstrated that low concentrations of progesterone are associated with reduced conception rates and underdeveloped conceptuses. However, on the contrary to that, high concentrations of progesterone early in gestation have been associated with increased embryo quality, increased production or secretion of interferon tau from that conceptus, advanced conceptus elongation or conceptus length, but ultimately leading to increased pregnancy rates. So if we dive a little deeper now into specifically what is the uterine function at this timing of maternal recognition of pregnancy. And certainly there are some changes that are induced by progesterone, because now progesterone is being prolonged, and there's certainly some changes that take place. And these being these secretions, from the endometrium. So we'll see that it maintains the endometrium functions that are necessary for the conceptus to continue to grow. Certainly during early gestation, there's a tremendous or exponential growth that is occurring just from day 7 to day 17. Also preparing for the process of implantation, placentation, but then all the way for development to term. And these progesterone-induced changes regulate expression of genes in the endometrium. But there is also kind of this little paradigm that the continual exposure of the endometrium or of the uterine endometrium to progesterone actually downregulates the progesterone receptors present in the endometrium. And this loss of progesterone receptors occurs before implantation. And this is very common in many species. And in addition to the progesterone effects on the uterine endometrium, there are some effects from interferon tau. And the main one being regulating the expression of interferon tau stimulated genes, or referred to as ISGs. And these ISGs are among the most upregulated group of genes, specifically in pregnant animals versus animals that are non-pregnant and would be normally cycling. Some common functions of interferon tau interferon stimulated genes would be nutrient transport for conceptus elongation, conceptus growth, but then also a localized immune system suppression. As now there's essentially there is a foreign object in the maternal environment, but that immune system has to be suppressed enough that pregnancy is maintained, but not to the point to cause any other illnesses. And it's also preparing the endometrium for implantation. So just to summarize our brief introduction here, really selenium deficient areas, so areas in the east and particularly in the southeast, necessitates the need for selenium supplementation to animals grazing in those areas. And we know that the conceptus produces interferon tau, which signals maternal recognition of pregnancy and therefore maintains the production of progesterone. And progesterone is necessary for pregnancy to be maintained. Both progesterone and interferon tau affect the uterine endometrium to prepare for pregnancy, but successful pregnancy requires a viable embryo, but also a receptive endometrium. And we mentioned a lot of these, but we still have some problems that we know of, right? So as we look or think just about the problems with selenium, so as we mentioned, the majority of forages and grains in the southeast would be deficient in selenium, necessitating the need to provide supplemental selenium. And as I mentioned earlier, research has demonstrated that selenium deficiencies in cattle have led to decreased immunity, growth, and fertility. But in addition to some challenges, with just selenium, we also have some problems that are occurring, Dr. Anderson, with pregnancy loss in cattle. And although fertilization rates are high, and they're actually nearly 100%, by day 30 of gestation, only 50 to 60% of those animals usually remain pregnant. And there's two periods of time that are usually classified or used to classify this percentage of pregnancies that are lost from fertilization. There's a time period referred to as early embryonic loss, which is fertilization to day 27, but more specifically, days 8 to 16, so the week leading up to maternal recognition of pregnancy, most of those losses occur, and it's actually 70 to 80% of all of the losses. But then there's another period of embryonic loss referred to as late embryonic loss, so day 28 to day 42, where there's still a small percentage of pregnancies that are lost during that time, but not as much as during days 8 to day 16. Which really leads us into our experiments. So really, the objectives of the experiments were to investigate whether the form of supplemental selenium, so the inorganic form or a mixture of the two, would affect the early bovine CL, the uterine endometrium, and the conceptus at the time of maternal recognition of pregnancy. So I won't go through, as we start to move through the experiments in a little bit more detail. Experiment 1 really focused on the effects of selenium on the CL at day 7 of the estrous cycle. So for this study, we used non-lactating 3-year-old Angus cross cows. These cows were housed at the University of Kentucky Research Farm in Princeton. And the treatments, or the vitamin mineral mixes, that being the inorganic form or a mix of the two, were supplemented to the animals through in-pasture kaolin gates, where these cows had an electronic collar around their neck that allowed them access to only their mineral feeder, where they had access to their assigned treatment. And for both of these experiments, we followed a very similar experimental timeline. These animals were subjected to a period to deplete their whole blood concentrations of selenium for 45 days. That was followed by a repletion period to bring these animals back up to a selenium adequate status for 45 days, but only with the inorganic form of selenium. Then those cows were randomly assigned treatment to either the inorganic form of selenium alone or the mixed form, or the mix being the inorganic and organic selenium combined. Cows were then set up to come into estrus, or come into heat, and 7 days after estrus, we performed a lutectomy, where we removed the corpus luteum from the ovary from the cow. So for this experiment, we collected whole blood all the way throughout the experiment, just to monitor or determine the total whole blood concentrations of selenium, and really to validate our experimental timeline, that we were depleting the concentrations of selenium, bringing them back up to a selenium adequate status, but also to make sure there was not a difference in those concentrations of selenium between treatments. And as I mentioned, cows were randomly injected with PGF2 alpha and monitored for behavioral estrus, which we used as day 0. And then on day 5, 6, and 7 of the estrus cycle, so these cows were not inseminated, so they were just normal cycling cows. But on days 5, 6, and 7, we determined the diameter of the CL using transrectal ultrasonography. And we also quantified the concentrations of progesterone in serum. Again, the CL were removed on day 7 via supravaginal lutectomy. And we also performed microarray analysis to evaluate the transcriptome, or the genes that were present in the CL tissue, the luteal tissue. And we used real-time PCR to corroborate those findings to determine the relative abundance of both cholesterol and immune-selected transcripts. But before we dive into the results from experiment 1, so this image here would be taken directly from the screen of the ultrasound while we were scanning these cows. And this, Dr. Anderson, is a very, very nice CL that's just under 30 millimeters in diameter. Again, so this slide here would represent the concentrations of total selenium in whole blood. So on our x-axis, we have our days of our experiment, and on our y-axis, the concentrations of selenium. Really here, just want to demonstrate the model of the depletion and repletion periods, but also to note that the whole blood selenium was not affected by treatment. And as we really start to dive in and move towards the concentrations of progesterone, and I apologize that this table is very busy, but I could not think of a different way to display this data. But I wanted to go back. So I mentioned in the very beginning that previous research from our lab had found that the mixed form of selenium had cows and heifers with increased concentrations of progesterone on day 6 and day 7. So that would be the results from year 1 and year 2. And in both of these years, there's about a 1 nanogram per mil increase in those animals supplemented with mix compared to those animals supplemented with the inorganic form of selenium. Now for this experiment 1, which really for us is year 3, I do want to point out that we had a reduced or a lower number of animals used in this study. But I do want to highlight that on day 7, although it's not statistically different, we do have about the same difference of a 1 nanogram per mil increase in those animals supplemented with mix versus ISE. And it's likely that this difference was not detected to be statistically different simply due to the reduced numbers used in this trial. The other thing I would like to highlight here is, although we have this numerical increase in the concentration of progesterone on day 7, that was not due to simply the size difference in the CL. Those CL were very similar in size in terms of weight and in terms of their overall diameter. But what got to become very interesting was when we looked at the results from the microarray analysis, when we started to look at the entire transcriptome of the luteal tissue collected from these animals. And from the microarray analysis, it identified that 887 gene transcripts were functionally annotated, but were statistically different with p less than 0.05. And actually, about half of these were up and down regulated respectively. However, there was no treatment effect on the steroidogenic enzymes. So there was no treatment effect on those enzymes that are responsible for the conversion of cholesterol to progesterone. But we have seen an increase in the concentrations of progesterone. So it's not an treatment effect on steroidogenesis, so it must be something else. And what we found was there were treatment effects on cholesterol biosynthesis and the immune response pathways. So as we start to look here at our concept map, and this group of pathways here in the bottom left-hand corner in yellow are all pathways that are associated with cholesterol biosynthesis. And if you remember from our introduction, the cholesterol biosynthesis would be one of the 4 sources of cholesterol for steroidogenesis and for the production of progesterone. But in addition to this entire group that is associated with cholesterol biosynthesis, we have another very large group of pathways that were found to be different between the mixed and inorganic supplemented heifers that are all associated with the immune response. And there's actually a common pathway that links those associated with cholesterol biosynthesis and the immune response elements, and that being the LXR, RXR activation pathway. And on the next slide, we'll dive into a little bit deeper detail on what transcript that might be. So as we really dive into the nitty-gritty and look deeper at the results from the microarray, and then we corroborated these results or confirmed these results by PCR. And 2 of the transcripts that I'd like to point out is this HMG-CR. And there was a tendency from the microarray data that those animals supplemented with mix had an increased level of HMG-CR. Now, this would be the rate-limiting step in cholesterol biosynthesis, and so it would be very important to having increased concentrations of progesterone. Interestingly, as we look on the figure on the right, SRE-BF1 it actually would be in the LXR-RXR pathway that connects the biosynthesis and immune response elements, but it regulates the expression of HMG-CR. And in both the microarray analysis and in qPCR, those animals that were supplemented with mix had an increased relative abundance of this transcript expression. So just to conclude briefly those results from experiment 1, those animals supplemented with mix had an increased expression level of several key transcripts, although I just showed a few for time's sake tonight, that are involved in cholesterol biosynthesis and immune response elements compared to those supplemented with the inorganic form of selenium alone. But ultimately, these results suggest or really play a pivotal role in increasing the early luteal phase concentrations of progesterone. So for our experiments, we thought it was very logical to continue the timing. So now go from day 7 of a normal cycling, non-inseminated female to look at the effects of the form of selenium further and through in gestation. So the next logical time point for us seemed to be maternal recognition of pregnancy. And for us, we looked specifically at day 17. But we hypothesized that the form of selenium, so the inorganic form or a mixture of the two, supplemented to yearling heifers, would alter the gene expression in the bovine endometrium. That would lead to an advanced conceptus development, ultimately, was what our hypothesis was. So for this experiment, we used Angus cross yearling heifers from both university research farms. And instead of the treatments being supplemented to these heifers through Kaolin gates, where they had access to only their mineral feeder, we decided to provide our treatments, our vitamin mineral mixes, using a very common method in the industry, just using a plastic mineral feeder with a rubber lid, so they had free choice access to their treatments. Again, we follow the same depletion and repletion period. The animals were randomly assigned treatment for 90 plus days. But this time, however, on day 0, so on the day that we detected estrus, we inseminated these females, and they were then hauled to the University of Kentucky USDA meats lab on day 17 of gestation, where they were euthanized and we were collecting our samples. So for this experiment, we were really after was recovering a conceptus. And although we would ultrasound the heifers before we hauled them to the meats lab, that was really to determine if there was a Corpus Luteum present on their ovary. Certainly on day 17 of gestation, Dr. Anderson, I'm good, but I'm not that good, but cannot detect pregnancy on day 17 with an ultrasound. So we did not know if these heifers would be pregnant until we had their reproductive tract upstairs in the lab. So when these animals were killed, we immediately removed their reproductive tract, and that reproductive tract was placed on ice. Then we actually used an AI sheath, and we injected physiological buffer saline, or PBS, into the reproductive tract. But before doing so, we would remove the ovary that was bearing the CL. So for this example, the CL was on the right ovary. We removed the ovary, and then we used butcher's twine to tie off this contralateral horn, or the opposite horn, because at day 17 of gestation, that conceptus should be present in the horn that contained the CL. So we tied off this horn, we passed an AI sheath into the uterine body, and we flushed the uterine body with fluid, gently massaged that around to float the embryo out of the endometrial folds. And then we actually cut the very tip of that horn off and made a very, very small incision. We flipped inverted the reproductive tract where we were draining the fluid, and then ultimately collecting the conceptus into the petri dish. But again, not knowing if that animal is pregnant until we confirm that with the presence of a conceptus. And of course, not collecting a conceptus by performing this 3 different times, that animal was deemed non-pregnant. And I always think this picture is very interesting, but this would actually be an embryo that we collected on day 17. But you can really start to see just how long or how advanced the development of this embryo is. And in addition to recovering the conceptus, we were also collecting endometrium biopsies of both caruncular, which is what would fuse with the cotyledon during pregnancy, forming the caruncle, and also the intercaruncular tissues. So as we again start to look at the effect of treatment on whole blood selenium, again, really just to display that our treatment period of a depletion and repletion, that our model worked. but also to note that there were no statistical differences in the concentrations of whole blood selenium in those animals supplemented with mix or the inorganic form during the time of treatment. And as we start to look at the profile or the concentrations of progesterone in serum, on our x-axis, we have the days of gestation, so going from day 0 to day 17 when we were collecting our samples. But actually, the serum progesterone was not affected by treatment, but there was a day effect. But I do want to point out that although that we were not statistically different in this study either, we actually ended up with 6 and 6 viable, or what we consider to be a successful or pregnant animals where we recovered intact embryos. We do see this little bit of a blip in the concentration of progesterone between day 5, and day 7. So very consistent with some of our previous research. And from the PCR, and we have for a few of these figures, we'll have the endometrial tissue, so looking specifically at the caruncular tissue or the intercaruncular tissue, and then the relative abundance of the mRNA transcripts. And MX1 is a classical ISG or interferon stimulated gene, and it enhances the response to potential viral pathogens, but is also very involved in pregnancy recognition and uterine receptivity prior to attachment. And when we looked at MX1 for this ISG, we actually saw in the caruncular tissue that those animals supplemented with mix had decreased relative abundance of MX1 compared to those animals supplemented with ISE. And as I was performing the PCR and on multiple transcripts, on almost 50 different transcripts in these genes, I noticed a very similar trend that there was a decline or a reduction in those animals and the relative abundance of all of the ISGs that I looked at when the animals were supplemented with mix. And to be honest, I was quite worried about that, thinking that it would make sense if we're trying to enhance ultimately pregnancy success, that these ISGs, and if the conceptus we hypothesize that the conceptus would be longer, theoretically, we would think that the ISGs would also be increased. There's actually a temporal relationship. So over time, there is a kind of a peak expression of when these ISGs are being elevated. And as I mentioned earlier, this paradigm loss of as the concentrations of progesterone are increased, or as there's continuous exposure of progesterone to the uterine endometrium, we see a decline in the relative abundance of the progesterone receptors. And PGR would be the nuclear receptor for progesterone. And in the caruncular tissue, there tended to be a decrease in the relative abundance of the nuclear progesterone receptor in those animals supplemented with mix versus ISE, which really makes a lot of sense. From our previous research, we saw that increase in progesterone, early in the cycle around day 17. So if there's an earlier increase in progesterone, and we know from this paradigm loss or continual exposure of progesterone to the uterine endometrium decreases the abundance or expression, of progesterone receptors, this trend actually makes a lot of sense biologically. And where I think the results or the data started to get really interesting was when I looked at the progesterone-induced mRNAs, but specifically those that their function was conceptus nourishment. And I'd like to point out that the energy source for the conceptus would really at this time would be glucose, but the uterus, or that it does not perform gluconeogenesis, so it does not produce the glucose for that conceptus nourishment, so therefore that must be transported into the cell, or transported into the uterus to be used by the conceptus, So as we look at DGAT2 and compare that to MSTN or myostatin, really serving a major difference here would be the timing of when they are providing the energy source to the embryo. DGAT2, interestingly enough, is an energy source up to the blastocyst stage of development. So around day 7, let's say, where myostatin is after the embryo has hatched and has began to undergo the process of elongation. And what we found specifically for DGAT2 was there was a decrease in the relative abundance of DGAT2 in the caruncular tissue for those animals supplemented with mixed compared to ISE, the inorganic form, excuse me. But on the contrary to that, we saw the opposite trend, where mixed was increased, the relative abundance of myostatin was increased in those heifers supplemented with the mixed form of selenium compared to the animals supplemented with the inorganic form of selenium alone. And again, myostatin would be serving as that conceptus nourishment after hatching, so during the period of elongation. And DGAT2 would be during that up to the blastocyst stage. So two totally different time points, and the time point that would be more similar to where we were looking or have more relevance would be myostatin as we're looking at day 17. So that embryo, as depicted by the picture a few slides ago, has already underwent that process of elongation and is growing quite rapidly. So serving as an energy source here, so leading to theoretically, right, an increase in embryonic length in the animals, the heifer supplemented with mixed versus ISE, just off of the findings from this slide alone. And interestingly enough, that is exactly what we noticed. So in this figure, we have our two groups of animals, our two treatments, the animals supplemented with ISE here in red, and those in mixed in green. And on our y-axis, we have our embryo length measured in centimeters. And an individual dot represents an individual heifer or an individual embryo that was recovered and measured, where the black triangle would be the mean or would be the average for each of the two treatments. And on average, the mean embryo length for the heifer supplemented with the inorganic form had a mean embryo length of about 17.5 centimeters. But in contrast to that, and actually significantly different, those heifers supplemented with mixed had a mean embryo length of almost 26 centimeters. So about an 8.5 centimeter increase. But why is that important? And Dr. Anderson, research has demonstrated, or there's a lot of thought that conceptus length is thought to be indicative of the quality of that embryo. But in addition to the quality of that embryo, also the success rate of pregnancy. And there's also thought to be differences in the regulation of genes that are associated with larger embryos compared to smaller embryos that are associated with the successful development of that embryo or that conceptus all the way to term, all the way through gestation. So to conclude or go through the conclusions of experiment 2, our research demonstrated that those heifers supplemented with mixed compared to ISE had a decreased abundance of progesterone receptors and interferon stimulated genes, although we just covered a few of those this evening. Importantly, there was an increased abundance of mRNA encoding myostatin. And again, myostatin is known to increase glucose secretion into the histotroph or into the providing the nutrients for the conceptus, which ultimately leads to advanced conceptus development, which is what we observed in the heifers that were supplemented with mixed versus ISE. But collectively, these results really suggest that the timing or the onset or when maternal recognition of pregnancy really begins is shifted and perhaps occurs earlier than day 17 or occurs earlier compared to in the mixed heifers compared to those supplemented with ISE alone. So just to summarize collectively all of the experiments and the results from these experiments, a form of dietary selenium differentially affects the luteal and endometrial gene expression profiles and ultimately conceptus development. And the mixed treated animals did not have any differences in steroidogenic enzymes. There was an increased expression of transcripts involved in cholesterol biosynthesis, decreased endometrial progesterone receptors, increased energy for that elongating conceptus, ultimately leading to the advanced conceptus development. Certainly, for these projects, need to acknowledge Dr. Bridges, Sarah Carr, my lab mate, all of the staff at both of the university farms, and do need to point out the project's support from the tonight. I would like to take this time to thank Dr. Buller, Kevin Laurent, Dr. Van Valen, for all of their help and support and guidance throughout my entire time at the University of Kentucky. It certainly was very life-changing. I'm very grateful for the opportunities that they provided me with, and the challenges that they provided me with certainly allow for opportunities to grow, and very grateful for all of their help along the way. And also would like to extend a thank you to all of the agents in the state that I work with, far too many to list, and also the producers. Dr. Anderson, with that, I guess I'd like to leave everyone with this quote of "keep your mind open, but not your cows." And I'd be happy to entertain any questions that anyone might have.
Awesome. Great job, Ben. Anybody that has any questions, you can just type them in the chat. But I'm going to start off with a question, Ben. You used several terms tonight that might be a little ambiguous to a lot of our listeners. Transcriptome, transcripts, PCR, okay, and microarray analysis. Can you explain a little bit in a little more detail exactly what those are, what that is, and maybe the flow from DNA to protein to biology that might fill in a gap a second or two, and then just pick one of your transcripts and expound on it just a little bit.
Sure. No, I probably should have changed these slides a little bit more, but really the transcripts, or mRNA, would be, I guess, ultimately encoding for those proteins. Maybe a word that I should have used, Dr. Anderson, would have been genes, or maybe simply ultimately DNA. So we're taking the DNA, actually reverse transcribing that, or converting that back into RNA. So we're taking the DNA and kind of breaking it down, essentially, going from the RNA to then cDNA. So then we start to build that RNA back, but then looking specifically just at those transcripts, or those building blocks for the DNA, or for those proteins that we would be referencing.
Got a couple of questions here, Dr. Kreitz. First one from Chris. Ben, have you done any work with injectable selenium, and in particular Multimin?
Well, Chris, that's a great question, and certainly one that I think several of us have probably gotten in the past. And personally, no, I have not done any research with an injectable form of selenium. This was all provided as a loose mineral, so I cannot speak on any data or any research that has been published or taken place with the injectable forms of selenium. I wish I could answer your question.
Oh, sorry. Go ahead.
No, I wish I could answer your question, but don't have that experience.
Another question. Given a bag of mineral with mixed sources is $3 to $4 more than inorganic, is it worth the additional cost? So is the 50% selenium worth the cost?
Is it worth $3? Yeah, sure. No, I think that's a great question. So depending on the day you call to compare prices between the mixed form of selenium and the inorganic form of selenium, the last time I checked, the number that I got from the nutritionist was $1 difference. So when I work the math out, and when we work it out on the suggested intake, so 3 ounces per head per day, for that entire year, it was 10 cents more per cow per year to provide those cows with the mixed form of minerals. So yes, on a per bag basis, it may be several dollars, but on a per cow per year basis, when you break it all the way out on the recommended intake level, it comes out to be pennies. So really there, then I guess for that question, it would be, how much is an increase in conception or an increase in pregnancy rate? And that work hasn't been done about what is the difference in conception rate to a fixed time to AI, or what are the differences in pregnancy rates in cows that are supplemented with mixed versus the inorganic form. That would be, was something that I was very interested in, Dr. Anderson, and tried to put that data together to see what is that numerical boost, or what is the effects kind of on a whole herd basis from a conception or pregnancy rate basis. And then would certainly be able to answer that question. If there's let's say a 10% increase in conception rate and it costs 10 cents per cow per year, then I think that pencils out pretty well.
Chris followed up and said, great presentation. He'd love to see Multimin fund you to repeat your study. A question from David. Is there a period of time that more expensive mix can be fed and cheap mineral fed at a different time?
Well, these are some really great questions. And certainly are some things that we've thought about, and that's actually what would is more than likely coming down the pipeline in terms of future research projects of what are those periods of time that would certainly be necessary. As you know, it takes some time to do those projects, but perhaps the mixed form or the more expensive mineral could be fed for a period of time, plus or minus calving and into breeding. What would that be? Or I really don't want to speculate on that, but I think that is some research that will be conducted in the relatively in the near future.
Irvin Yoder, who you know very well, Ben, said great presentation. And then we had another question. Was there some earlier research? This is from Dr. Lynn Cooler, with mix showing improved embryos. Maybe Justin and I were involved in that.
I think you and Justin were involved in that, so I'll let you take that one.
No, go ahead.
No, from what I remember, yes. But did you also look at the quantity of embryos recovered? I think you were. Was that in superovulation protocols?
Yeah, it just improves quality quite a bit. Yeah. So on day 7, but those are time frames that are coincides with your CL collection, but certainly far different from the biology on day 17. But yeah, we did see an increase in embryo quality with form of selenium in embryo superovulation and embryo collection. Another question: was your research solely done on cattle, or were there any studies done on goats?
Yeah, so all of my research would have been solely on cattle, but both in cows and heifers.
I have one thing to ask you, if you know about some of the statistics, not the stats in your research, but a couple of the stats that you pointed out on pregnancy loss. From day 28 to 42, You talked about a 3 to 14% embryonic loss during the late embryonic period. I think we're actually seeing closer to 10 to 15 all the time.
Yeah,
So you got 20 cows and you get 18 of them pregnant. You're going to shed 2 between day 28 and 42. And I'll be it bluntly, I'm seeing that a ton in
I agree.
the work that we do. And so what would you propose, or I attribute it, I attribute some of that to a very small part to genetics, Dr. Bullock. But I try to attribute a lot more of it to disease, but maybe I'm looking at the wrong thing. Maybe
No, I
it's
think,
selenium.
yeah, I think you're right. You're definitely totally agree with you on that number being higher, certainly higher than 3%, at least here in the, very near past. I definitely agree. That number is probably 10 to 15%. I think, like you mentioned, probably a minimal impact of genetics occurring. Probably a lot to do with stress. I think there's a big disease impact, just off of some things that we've worked on, but also just kind of that, as some of this research really dives into, is that uterine environment, and kind of being that uterine environment is present during that entire time period, and those factors. So probably collectively being stressed, whether that's nutritionally, heat, or disease. I think it'd be pretty hard to pinpoint the specific reason for that increase during that time period.
Is there 2 waves of interferon production? I can't remember. I'm not quizzing you. I can't remember.
Okay.
But I thought there was a wave, starting around day 16, 17, kind of, and then it dips down and it comes back up. Is that right or wrong?
No, that's right. So there's a wave there in the beginning. There's kind of 2, and there is some overlap, but there is kind of a valley as well. So you'll see that increase early. That peak, 9 to 10-ish range, and then by day 17, so like for this study, those ISGs were all lower. The interferon stimulated genes were lower in the mixed animals, just from this time shift occurring. But then I think it's not as high as the early one. So maybe there's a little bit of an overlap there to kind of modulate those transcripts.
I've been blaming it on subclinical disease for years, but I'm curious if it's not more selenium. Or Dr. Lemcooler, do you have any thoughts on that?
No, I think, it's some of the challenge we deal with is not knowing the previous status on some of the cattle either. We know that some herds are certainly going to be more prone to deficiencies than others. And, I think in severely malnourished or low mineral supplementation type herds, that it's probably going to be higher, particularly given the suppressed immunity that, we talked about.
Need to figure it out because it's starting to aggravate me. 10% loss in that after a preg check on day, positive preg check on day 28 is really aggravating.
I still don't think we can completely discount those recessive lethals. I mean, I don't have an idea on the percentage, but they're definitely out there.
I mean, would you think like 5%, or I don't even know where to guess on that.
Well, for a particular time frame, I wouldn't either, Les. I think different ones are probably going to hit at different time points. I'm guessing total embryonic loss close to the 10%. just a
Yeah,
pure
I would agree with that number if you include the, day 7 losses and day 17 losses. But we particularly with the new preg check shoot side preg check thing that we've got. I've got a lot more people doing 30 days and then coming back at 60, and we're losing way too many pregnancies in that time frame. And it's something that something we need to look at, I think. Chris had another question, Ben. In your studies, the water source was well or municipal, not surface water?
That's correct. So all of these animals would have been maintained on city water.
Any other questions? Kevin, Dare, Jeff, anything to add?
So that water was the same water that Kevin drinks, right? So maybe there is something wrong with the water.
That's right. It's also the same water Jeff drinks, so because of the animals in Woodford. So it does explain a lot, Dr. Bullock.
Hey, be nice.
There you go.
Well, if there's no other questions, Ben, thanks a ton for presenting for us tonight and participating in our UK Beef webinar series. I think everybody can see on March 8th, we have another one. It's another one of our Shooting the Bull webinars where all the specialists will be together to answer any questions and to bring up or stir up any sort of hot topic type ideas. So hopefully we'll see everybody back on March 8th. And if not, we have one every month. So just if we can't join us on March 8th, look for us in April and May. So anyhow, thank you, Dr. Kreitz. Appreciate your time, and thanks everybody for joining us.